All right, so this is remediation 1, remediation cell cycle. Right. So you were given a task. I said, here you have this cell. You have this cell that has 2 chromosomes. I'm looking at the nucleus right now. And the 2 chromosomes, one came from its mother, which had a big A. Mother gave this child a big A, and mother gave this child a big B. Where this creature's father gave this child a small a and a small b. It's a cell, so it looks something like so. There's a nucleus. And I asked you to take it through the cell cycle. One would hope that you would know that the very first thing the cell is going to get triggered on is to go through G1, where it's going to increase in size. The nucleus is not going to change. The nucleus doesn't change, but the cell gets bigger. And that means that if you had endoplasmic reticulum and Golgi body and ribosomes and mitochondria, you doubled or tripled the number of mitochondria. The endoplasmic reticulum doubled or tripled. The Golgi body that doubled or tripled. The ribosomes doubled or tripled. Everything became more and the cell became larger. For those of you that are taking the quiz today, I think that's everybody, you should know that right before you go from G1 to S phase, there is a checkpoint. Arguably the most important checkpoint number 1, is right here, right before S phase. If the cell has enough material and it's healthy, it has enough nutrition, and it's big enough, then it goes on and goes into S phase. So those are kind of the things and I'm not writing these things down, but you should have them written down or somehow demonstrated that G1, the cell grows, that the nucleus is the same, and then here in S phase, what you got is basically a copy of what's going on in G1, except now can anybody tell me what's going to happen now? Sorry, what? That's right. Now we're going to get into DNA replication. So now the nucleus is going to, there's something interesting happening in the nucleus. Where in G1, there was nothing really happening in the nucleus. Now we're going to look at the nucleus. Now when this doubles, when this DNA doubles, I'm going to have to draw it a little smaller here. Somebody said, well, isn't it going to have an X? And I said, yeah, well, yeah, in a way. What's happening is that these are compacted. They're doubled first, and then they're compacted. And so what you get is something that looks like this when you look under the microscope, and you'll see that today because we're going to have our lab today. And it's doubled, right? So then you have the second one looks like that. And you keep them together so you don't lose them, so they don't get mixed up with a centromere. And since these are sister chromatids, and I would expect those labels, since these are sister chromatids, what I expect to see is that this is a big A, this is a big B, because that's what they got from their mother. This is also big A and big B. These are identical. Sister chromatids are identical. Then again, because they're compacted, they're not unwound the way we had it drawn previously. This chromosome from the other parent is also doubled. It also has a centromere connecting it, keeping it together. And these are also identical, but they're identical to each other. So they're little a, little b. So that is what's going on in S phase. You're just copying it. That's it. That's all that's going on. It's been doubled. The DNA's been doubled. Of course, that would happen in humans with 46 chromosomes. So there'd be 92 copies of the DNA after all this is done and said and done. The next thing is you're going into G2. And in G2, the only thing that's really is going on, you're not really getting that much bigger. You're just checking to see how it all went. You're making sure that there's no errors. If there's errors in the duplication of the material, if there's errors, you just got to check. So what does that suggest that there is in G2? Right before you go into a checkpoint, that's right. Let me make it bigger. I'm going to make it just slightly bigger. It might have gotten a little bigger, but not much. So you have another checkpoint as you go from G2, where you checked everything, to M phase. It might have gotten a little bigger, but in general you have another checkpoint. This is checkpoint 2. Not as critical as checkpoint 1, because at checkpoint 1 either you went through the cell cycle or you didn't. So this really was probably the most important checkpoint. But here also you can pause. Remember, what are your two options? Pause. The checkpoints have two options. 1, 2, and 3. You either pause, prepare, or die. We call it apoptosis. Okay, so I hate writing with my finger. Apoptosis. So now we're in M phase. M phase is pretty straightforward. I'm just going to draw out, I'm going to do this, I'm going to put a dotted line for the cell. And what am I really focusing on in phase? M phase, what am I focused on? The nucleus. So that's what I'm going to focus on here. I'm going to draw out the nucleus. Well, now we're going to go through the 4 phases. We got prophase, we got metaphase, we got anaphase, we have telophase. And then after mitosis, this is mitosis. Right after mitosis, you have cytokinesis. I'll just put C. In prophase, you have a real condensation of the nuclear of the nuclear material. You have a doubling of the centrioles, which are going to act as anchors for the spindle fibers. The spindle fibers start to form, right? In prophase, and also the nucleus disappears. Now this is important. The centrioles also double, but they also move to opposite sides of the cell. And that allows for the nuclear material to be to be separated. That's going to become important because you need a set of nuclear material on each side of the cell. So then the spindle fibers form. Right? They start to form. The centrioles they've been doubled. They're on opposite sides. The nuclear material's gone. All that happened in prophase. In metaphase, the chromosomes line up. They line up with the sister chromatids facing each opposite direction. The homologs line up. Now, why does that make sense? Why does it make sense that they'd line up in this direction? It's what? Yeah, it's easier to split. You're gonna make sure, because what are the sister chromatids? They're exact copies of one another. So let's remember, you have a big A. You started with a big A from mom and a big B from mom. I think I said that mom. It doesn't matter at this point which who it was from, but. And the other one was a little A from dad and a little B from dad. All right? So you started with, in the very beginning, if we go back to the very beginning of the problem that you were given, you had, what did you have? What was your genotype? Big A, big B. I'm sorry, big A, and big B, little b, correct? Yeah, that's your genotype. So if you're going to end up with an identical cell, you have to have a big A, little a, a big A, little a, and a big B, little b. Because you lined them up this way, what is each new cell going to have? Big A, big B, little b. Because this is going to go this way, that's going to go that way. Just because they're lined up the way they are. That would be the drawing of metaphase. Now anaphase, should I draw it? I wonder if I should draw it out, or yes, I should. I was going to just erase, but. So that was prophase of mitosis. Now we can look at metaphase of mitosis. And in metaphase of mitosis, they're lined up. Oh, come on. Metaphase of mitosis, they're lined up. And then in anaphase, what happens? They split, that's right. Now in anaphase, now you're splitting. Now you have one going in that direction. I should draw it this way. One getting pulled in that direction, and one. They're actually pulling themselves with the kinetic core. They have these things called kinetic cores in the middle of the centromere. These kinetic cores are what are eating through the spindle fibers and moving the sister chromatids to opposite sides of the nucleus of the cell. As you can see, they kind of look like they're zooming along. Remember that means that this again is big A from mom and big B from, and this was a sister chromatid, so this is big A and big B, and of course this was little a and little b, and this was little a and little b. So now in anaphase, you can see that they're right. Not just are they separated in number of chromosomes, but they have the right combination of chromosomes in each side of the cell. This is happening in anaphase.
So anaphase when they separate.
When the chromosomes are separating. Now, in telophase, now in telophase they're separated. So I'm going to go ahead and. So they said, oh, come on. So now you have your cell membrane still there, never went away, because if it did, the cell would die. So now in telophase you have this cell, I'm not going to draw the organelles. I'm just going to draw the one chromosome that has the from mom, and the one chromosome from dad, the one chromosome from mom, and the one chromosome from dad. So to, again, to emphasize big A, big B, and big A. Big B, remember each chromosome can have many genes on it, but we're just simplifying it here. Will have many genes on it, all right? So what's going to happen in telophase? You've moved them over. You have a set of centrioles on each side, the way you started. You have the right number of chromosomes on each side. They're the right type. What is going to happen now?
You're gonna have two nuclei, they're gonna form and
That's right, you're going to form a nuclear membrane. That's right. Somebody mumbled something over here as well, but I couldn't hear it. I'm sorry. So the cell membrane stays the same. I just have to expand it because I didn't give myself enough room. So what happens is you start to form a nucleus, two nuclei. So now you got your nucleus is back. But now you have a cell with two nuclei. Right? Two perfect. What is the next step? And still in telophase, this is all happening in telophase. What has to start happening? Cytokinesis. So now you're done with mitosis. Now you have to start doing cytokinesis. So in cytokinesis, you start to pinch off, the cells start to pinch off in animals and other living things without cell walls. They start to pinch off. You still have the nucleus. Let me just try to copy this. Uh-huh. They do, but it's a process. It doesn't just happen, right? So you're right. They're going to end up fully dividing, but first they start to pinch off. They have to pinch off, otherwise you don't. Does that make sense? Yeah. So they start pinching off so that they can fully divide. So they start to pinch off, and then of course, as you just suggested, they pinch off even more. And then finally, what happens? Somebody just said it a minute ago. Finally, they just completely close off from one another, and now you have two cells that are identical. Since you doubled the stuff in G1, they have exactly the same amount of stuff. Are we all good? That would be mitosis, and then meiosis is a little more complicated. I'll record meiosis for you when I do this with tomorrow's kids.
So I would say inside of these have like together.
They're both part of M phase, but mitosis is a division of the nucleus. So M phase includes mitosis and cytokinesis. All right. Are we all good?
Yeah.
So I just gave you at least 100 points there, as long as you wrote it correctly. And if you want the other 100 points, get meiosis straight. Yes, you can say it. You're going to do it all again, but there's differences. Like, what are the differences between mitosis and meiosis? So it does not go through S phase the second time, so you don't copy the DNA again. So when you go the first time in meiosis 1, instead of having them line up this way, they line up as tetrads. All right, if you want to know about meiosis, you come over here. I'll go through it. So otherwise, you can just turn in what you got.
Mr. Mendoza, do you mind, so I wrote mitosis like an example here, but I didn't write it in the cell cycle. So for this, I wrote the cell cycle.
That's fine. Just staple them together. Make sure your name's on it. Make sure everything's labeled and it's clear. If I'm confused, I can't give you points. So. It's a good question. It's obviously has to duplicate some material. Make sure you have your period or class on the paper. I don't want to have to search where you're at. So look, whoever's doing meiosis, if you want to see this, what happens is the big differences in meiosis is you don't do S phase again. You do G1, you grow some things, you make sure you check everything, then you go into M phase. Okay? Yeah, that's right. They're going to divide twice, but you don't want to divide the DNA because then you'll end up, all you're doing is mitosis twice, and that's not good enough. You want to half the DNA. So to half the DNA, you don't synthesize it twice.
Minus 2, sorry.
All right, no problem. So let's take a look at what happens in meiosis. So in meiosis 1, in meiosis 1, instead of the lining up during mitosis of meiosis 1, during prophase, first of all, I should say, in meiosis 1, you have what's called a tetrad forming. Tetra means 4. So what that means is, if we look at it, what that means is that you have the little a and the little b, and the big A and the big B. Just put it there. You don't need to talk to me. You have a tetrad forming in prophase 1. So one of the big things that happens in prophase 1 is that there's a switching of DNA. Randomly, a piece of these two chromosomes will switch. So what you'll get is some of mom's DNA being mixed with some of dad's DNA. So instead of big A, little a, big B, you have little a, big B here. And on this one, instead of little a, little b, you have a big B here.
Wait, okay,
So now you have 4 different combinations. You have big A, oh, I put big A there twice, that's a mistake. You have big A, big B, which was original. You have little a, big B, that's the mixing up. The crossing over happened, that's what crossing over. You have big A, little b, right? And little a, little b. So you literally mixed up the DNA. That's why we call it recombination.
So I see that there's 4 of them. Does that mean you're separating the sister chromatids?
Excellent question. No. So in mitosis, you separated the sister chromatids. These questions would be great in class. Will be great in class, should be great in class. Ask them in class, okay? In mitosis, you separate the chromatids, sister chromatids. In meiosis, you're separating the homologs. So these are staying together. So now during M phase, or during metaphase of M phase, right? During metaphase of meiosis 1, so metaphase 1, what happens is that the centrioles still divide, they still separate, spindle fibers still form, you still get kinetic cores, but now these things, instead of separating the sister chromatids, now you get,
Is it time for class?
I don't know. Now you get this chromosome, this homolog. I shouldn't do green, let's do red. This homolog moving one direction, and this homolog moving in the other direction. So you get this one moving this direction and this one moving in that direction. So they separate as a pair. Does that make sense?
Are there names for these cycles, or just called meiosis one?
Meiosis 1. And then metaphase 1. This is metaphase 1, and then anaphase 1. And then telophase, after telophase and after the first cytokinetic event, what you have are two cells, right? Think about what you're going to end up with because you did this. If you think about what you end up with, there's no doubt you know what you're talking about. Come on. At the end of the first cytokinesis, what you get is this pair of chromosomes that went through crossing over, correct? So you have one set that's still big A, big B, one chromosome still big A, big B. Then you have another chromosome, though, that's big A, but then little b. And you have another one that's over here. Oh, I'm sorry, I messed that up, didn't I? Yeah, that's right. I didn't do that before. I think I did little a over here.
Do they still have checkpoints again?
Yeah, you still got checkpoints. That's still happening. You have little a, big B, and over here you have big A, big B, and over here you have a big A, and then you have the little b. Does that make sense?
Yeah.
I messed that up. Does not make sense. This should be little a, little b. I don't know what you're doing. Whatever you're doing, just do it. I have to finish this thought. So what you end up is big A, big B, little a, big B, little a, little b, and big A, little b. So you have 4 different options. Now you do mitosis. Meiosis 2 is just mitosis. Now instead of lining up there's no tetrads, no crossing over in meiosis 2. They line up on the metaphase plate just like they did in mitosis. But now you only have half the DNA because you didn't do S phase. Do you remember that?
Yeah.
So because you did half the DNA, now you end up with 4 cells, right? Because each one of these is going to divide, right?
Yeah, so after meiosis, then they go through mitosis again.
Well, no. No, they just do meiosis again. But no, it's just like mitosis. Meiosis 2 is just like mitosis. But there's no double of the DNA. So now you have one gamete, these are sex cells, with big A, big B. That's the original form, right? Shush of one of the parents. One that has little a and a big B. And one that has a little a, little b, and one that has a big A, little b. So now you have this recombination, right? This different set of DNA. Instead of 2 different options, big A, big B, little b, you got 4 options. That's why you have a Punnett square the way you do. That's why there's 4 spots in a Punnett square, because there's 4 possible combinations in between 2 heterozygotes. Does that make sense?
Yeah. Do you
All right.
want, is it okay if I finish this? Just like finish writing down what you said, or you just want me to
No,
turn it?
just turn it. Whatever you did. All right?
Can I have to stapler?
Yeah, staple it and go. All right.
Was it here?
Huh?
Here?
Yeah. So that was half an hour, perfect timing.